Hello everyone, and welcome to the IFST and Food Authenticity Network webinar on food analysis for non-analysts. My name is Selbrani Elahi, and I'm the Deputy Government Chemist and Executive Director of the Food Authenticity Network. It's my pleasure to chair the first part of this webinar, and I'll be joined by our first speaker as the co-chair to the Q&A session. Food analysis, as you probably know, is a discipline of dealing with the development, study and application of analytical procedures for characterising the properties of food and their constituents. The aim of this webinar is to give practical guidance for stakeholders who commission analytical work and to give an overview of where you can find information on analytical procedures commonly used. In addition, we've brought together experts from 3 food authenticity centres of expertise who will describe analytical innovations that have practical applications in the food industry. The webinar is being recorded and will be uploaded to IFST's Webinar Hub. Please feel free to make comments and ask questions throughout the webinar using the Q&A button at the bottom of your screen. You can also upvote other people's questions by clicking the like button next to their question, and this will help show us which are the most popular questions. There will be a Q&A session after the presentation where we will respond to as many of these questions as possible. For more information on our speakers, please look at the webinar page on the IFST's website for their biographies. Before I introduce our first speaker, we'd be grateful if you could please answer just one question in a poll. What we'd like to know is, does your organisation have a written food analysis strategy? We'll give results to this poll just before the Q&A session and talk about it during the session. So it's my pleasure to introduce our first speaker today, Sterling Crew, who I think doesn't need much introduction to most people. He is the President-elect of the Institute of Food Science and Technology and also the chair of the Food Authenticity Network's Advisory Board. Sterling has 35 years of experience as a regulator, retailer, brand owner and food manufacturer, and has given him a unique perspective on the challenges of food safety and governance in the global food supply chain. This webinar is based on one of his papers, and you have a link to it in the resources section of this webinar. And that's all from me. I'll hand over to Sterling. Sterling, please.
Thank you very much, Selvrani. So hi guys. So as Selvrani has said, this is food analysis for non-analysts. And it really comes out of a conversation I had over coffee with Selvrani and Michael Walker, who we all know as one of the great public analysts from Queen's University, Belfast. And the conversation was around Michael saying, you know, when I speak to a lot of food scientists, sometimes when they are commissioning work, analytical work, they're not quite sure why they're doing it. So sometimes we don't have a strategy for doing it. And other times they're not quite sure of the questions to ask. And then he was also saying, and sometimes when you give them the answer, they had trouble interpreting what the answer actually says. And that really resonated with me. As I said, I've been actually in the food industry for 40 years. And I worked as a regulator. I worked for 2 big retailers. I worked for one of the biggest brands in the world. And I worked in manufacturing. So in my career, I probably commissioned thousands of tests. But really, I started to think about what Michael has said, and did I really understand what I'm doing? Because this is a really, really specialized area. And perhaps the tone of what we're going to be talking about is, look, there is help out there, guys, and there are techniques you can use to maximize getting that right information that you need for yourselves and your business. Next slide, please. And so food analysis, even when you think of it, you try to put it into one bucket. Oh, there are food analysts. But in fact, there are many types of food analysts, and there are many types of analysis. And you can get specialisms in each of these. You know, the work that goes on in GMOs, we've seen attention in allergens, we've seen attention on origins, authenticity, fraud, safety. So there are lots and lots of reasons for doing testing. And each of those might have its own expertise. And so I think you need to make sure you're speaking to the right expert in that particular area that you might want to get some information on. Next slide, please. I would urge you to join the Food Authenticity Network. Well, Silver and I would say that, but it is a great resource, a free resource. It was set up actually after Chris Elliott's commission on the meat scandal, where we realized that we didn't have the appropriate tests ready or know which labs could do what. And so this has been set up. It's an open book resource. You get it online. There's centers of expertise, so you can click into that, either academic, general proficiency. There are 9 of those, specific techniques and specific commodities. So we have areas on that. And if you're looking to do a test, you can find out what test to use and which laboratories can offer you that service. You actually get a named person, you get their email, you get a telephone number. So it's a really quick fix, amongst other things it does on horizon scanning. Next slide, please. And there's the reference of the official laboratories, guys. Don't forget that. And what I'm really doing is signposting to you where these resources are. And as I say, if you look in the resources section of this webinar, you'll see all the links to this. And there's the National Reference Laboratories from the Food Standards Agency and FSS. There's Feed and Food Official Laboratories, and obviously the organisation which the government chemist, where Selvarani is the deputy chemist. Obviously, they do a lot of great work when it comes to setting standards and almost being the honest broker when it comes to analysis. A great resource, I think, is really underused. Next slide, please. And as I say, there's the Food Authenticity Network. And we have all this research on kind of research reports, standard operating procedures, surveys, scientific opinions. And it's great for horizon scanning as well. So it's not just about analytics. If you want to know what you should be looking out for and how you can actually find out and validate your views on your supply chain, it's great for that. Next slide, please. And a shout out also to the IFST. And one of the great things about the IFST is it has an immense amount of knowledge base in there. One of the difficult things, sometimes it's really hard to find out where it is. And one of the other reasons that sparked my paper is I came across 3 information sheets that they produced. Here you'll see all 6 of them. And I thought, wow, this is fantastic. If I'd have read this 30 years ago, my life would have been a lot easier. So I'm giving you links to these as well. And I really urge you to meet them. And really the paper that I've done is a synopsis of this along with my personal opinion. And honestly, the failures I've had in 40 years, I've tried to include those where I've made assumptions. And you know what they say about assumptions? They make an arse out of me, not you and me. If you make assumptions very clearly, you can get yourself into trouble. Next slide, please. And so here is the paper. As I say, grab yourself a cup of coffee. I hope you're members of the IFST. If not, join. It's a great organization. It's about 6 pages long. It's written hopefully accessibly rather than an academic paper. And you can get it online. And I think Robin has kindly set up a screen there so you can actually go to the online article, again, very free. It's a great resource, the IFST's backlog of great papers. Next slide, please. So we come to the real nitty gritty here about what are the important criteria in selecting analytical techniques, and more so of selecting your partner. And this is partnership. I mean, you can treat analytical work as almost a commodity, that you go to the cheapest person and you're trying to control price. Cost is really important, never more so perhaps than now. Or you might go for a whole basket of reasons, a kind of almost a bespoke service, what you want. And so if you're selecting your center of excellence, your center of expertise, your partner of choice, however you want to describe your partner, now here are some of the considerations, and there's more information on each one of these. You know, how precise is the work they do? Can it be reproduced? Are we seeing reproduces? You know, how reproducibility is it? What's the level of accuracy? To me, it's things like speed as well. People forget that. Now, if you've got a laboratory and you have a major problem, maybe a crisis on your hands, you might be facing a withdrawal, and then the analysis is really critical. You want that laboratory to be able to turn that result round, you know, almost within hours, not within days or weeks. So, having an agreement about exceptional testing is important. As you say, you have to be aware of their sensitivity, of the equipment. I always look for what type of third-party accreditation that they have as well? Do they have a 9001 actual accreditation for the management system? And do they have accreditation UCAS for each particular method? So that's the way that I start to build confidence. And certainly when I was in manufacturing, what I try to do is to say to my retail customers, look, don't trust me, because I would say what I would say, go to, trust this respectable laboratory. They're third party. I can't influence their results. I can't influence their interpretation. I can't influence their assessment of the facts. And I think that helps give the retailers confidence. And as a retailer, when I saw that, that gave me confidence. And in brand management as well, you wanted to check your work. So even working for the biggest food company in the world, we were obsessed with getting outside people just to validate what we're doing. So that's really important, I think, more than anything else. If you take away from this, you're a commissioner. Have you got the right partners? And it might be a partner, or it might be a couple. As you saw from the kind of opening slide, certain people are better at certain techniques than others. Next slide, please. I think one of the shocking things for me is how testing sensitivity has increased over the last decade ago. Before we were talking parts per million, and now we're looking at parts per billion, or even parts per trillion. And that has created a problem for us because now we're finding things in our food that we didn't know was there. And we need the laboratories to interpret that information once they find these things and to put it into context. After all, as a monk said in the 17th century, the poison is in the dose. And so that interpretation is really important. Next slide, please. And just as a summing up, it's essential to select that laboratory that provides the expertise, the confidence in its instrumentation and software. And it's not always laboratories that are doing the analysis. I mean, when I started off many moons ago, NIR was in a room and you had a PhD student operating it now, and it cost you thousands and thousands, hundreds of thousands of pounds. Now you can buy one that fits in your hand. It costs you 200 quid, and you don't need to have a PhD to operate it. You can just train someone to do it within half an hour. So I think not only will there be a lot more analytical work that produces more results we didn't anticipate, I just think the level of analysis will go up exponentially over the next couple of years as some parts of it are almost de-skilled and made to be very, very cheap. Of course, when you do these results, you absolutely need an expert to interpret them. And, if you have a failure, then you want to make sure that it is tested by an expert to make sure there's not a problem with your equipment. Next slide, please, Ronnie. So these are some of the performance characteristics that you use to assess if the method is fit for purpose. And that's one of the big lessons working with the Food Authenticity Network and from people like Selvarani, who I class, an expert chemist, an expert in analysis. And I've learned lots of lessons about limits of detection, limits of quantifications, the selectivity of the test, how sensitive it is, how linear it is. And for me is the uncertainties. I have taken action past perhaps on analysis where I didn't really appreciate as much as I should do the level of uncertainty that's within that. And again, big lessons for me from Michael and Selvarani about the interpretation of the results and what the tests you can interpret from and what you can't. And the last slide, please, as I just come to a summation. So this is a process. And it is not just about somebody in a laboratory sitting there and doing the analysis. There is a beginning to that, and there is an end to that. And the beginning for me is, well, why are you doing this analysis? Analysis is not a QC function, which I thought it was for many years, to be honest with you. It is not a QC function. I think it's a strategy function in your food safety management system. But did I have it as a strategy in my food safety system? I know we've got a poll on it and we'll ask for your honest opinion. I don't think I had for years. I always thought it was QC. You need a sampling plan so that you're actually having enough of your product that it means something statistically significant. the collection of the sample is so critical. I was having failure after failure after failure on a test, and I couldn't work out why. It seemed counterintuitive. And then when we did our root cause analysis, we found it was the product was not homogeneous, and the way it was being collected actually was not giving us a true representation. You know, I live out here in deepest rural Wiltshire, and we have a saying here that always resonates with me. You know, you can't fatten a pig by weighing it more often. And sometimes if you do get into analysis, it's like, well, I'll just keep measuring it until I get the answer I want. And that's the very last thing you do, as you know. So it should really drive your analysis, should drive behavior. Even if you just keep getting the same results all the time, you should question yourself why, let alone, well, I'll just keep testing until I get the right result. And the sample preparation as well. In my career, I've seen lots of mistakes being made by the way that samples have been prepared. And then it's the analysis and the report. Does the report really answer the question that you asked? And do you understand what it's saying and interpreting that report and the weaknesses within that report, so that the final thing you get from this is you can make an informed decision, you can take action, you've got confidence. But I think unless you have these elements within your organization, whether you're in retailing, hospitality, whether you're manufacturing food, you know, whether you're in the supply chain, in agriculture, looking at pesticide or insecticide, or, you know, whether you happen to be a processor somewhere in that chain, for the whole supply network, these principles have got to be driven. And, you know, sampling does have its weaknesses. So there are other ways we should be addressing food safety and authenticity, but this is a good tool that's in your toolbox that you can use. And certainly when it comes to authenticity, sometimes it's the only way, if you've got fraudulent behavior in your supply chain, that you can actually find the problem. And maybe again, going back to the horse meat crisis, that was somebody in Ireland doing some species analysis. Even though there were audits and, you know, paper trails and all the other things, in the end of the day, what picked up the authenticity was analysis. So it does have an important part to play. So guys, that's just my view. And as I say, we've got some real experts in here. And if there's any message I have learned is know your limit of expertise. Find the expert, find out where the people who can help you are, listen to them, make sure you've asked the right questions, got the right information. And I think you won't make the same mistakes that I used to. Thank you, Silverani.
Thank you so much, Sterling, for being so candid about your career and your experiences throughout your 30 years experience. We'll come back, and Sterling is obviously going to co-chair the Q&A. So if you've got questions for Sterling on his talk, please start putting them in the Q&A and we can pick them up later. So it's my pleasure to introduce our second speaker today, who's Dr. Malcolm Burns. Malcolm is the lead for LGC's Food Authenticity Centre of Expertise. Malcolm is the principal scientist at LGC in Teddington in the UK. He has over 20 years postdoctoral experience as a molecular biologist and is the manager of the UK National Reference Laboratory for GMOs in food and feed. So with that, I'll hand over to you, Malcolm, to talk about seeing is not always believing, multi-spectral imaging, MSI for food screening. Over to you, Malcolm.
Good afternoon, everyone, and thank you for that introduction, Salvarani. As Salvarani said, my name is Malcolm Burns. I'm the head of the GMO Analytical Unit and principal scientist at LGC, and I'm going to give a presentation this afternoon entitled Seeing is Not Always Believing, the Use of Multispectral Imaging for Food Analysis and Screening. So my area of specialization traditionally is with nucleic acid or DNA molecular biology methods for food authenticity testing, so inclusive of real-time PCR, digital PCR, next generation sequencing. But I'm here this afternoon to talk about the use of multispectral imaging for food authenticity. Now, in terms of definitions for food authenticity, there's a number of definitions available for that, but within the scope of this afternoon's 10-minute presentation, what I really wanted to look at in terms of authenticity was, is food what it says it is on the label, and has the food been adulterated or substituted? And with that in mind, I'm going to give 4 illustrative examples of how we have applied multispectral imaging for food authenticity testing. So, what do we mean by multispectral imaging? Well, multispectral imaging uses the simultaneous measurement of 2 things. This is reflected light across a broad range of wavelengths and an order of spatial magnitude to allow fast and accurate determination of surface color, texture, and in some instances, the chemical composition of ingredients in a sample. So for example, if we take traditional red, green, blue imaging, this ranges from 400 to 700 nanometers in range. But with multispectral imaging, this can range from 230 to 1050 nanometers, or from the UV through to the near infrared spectrum there. We've got an example of one of the instruments we are using at LGC in the bottom right-hand corner there, which is the VL4 multispectral imaging instrument produced by a videometer or videometer. So how does multispectral imaging work? Well, it really uses reflectance from 19 different LEDs to build up a unique spectral profile, almost a unique spectral fingerprint of a sample based on a composite picture. So as an example here, if we take a cross-section of a Petri dish, which we can put the sample in, we're basically looking at the reflectance from one LED on that and measuring the reflectance there. But of course, the Petri dish itself is not just composed of 1 pixel. It actually composed of 2192 times 2192 pixels, or 4.8 million pixels. Now with multispectral imaging, we're also not just shining one LED on a pixel, but 19 different LEDs on there. So each pixel measures the reflectance from 19 different wavelengths. And each sample therefore consists of up to 91.2 million data points, which gives it a very high discriminatory potential. So without further ado, I'll go on to one of the first examples of where we've applied multispectral imaging, and this is with wheat adulteration or pasta adulteration. So pasta needs to be made from durum wheat, Triticum durum, which is typically a high quality but low yielding type of wheat. But there are some unscrupulous individuals out there who will bulk out their durum wheat harvest with common wheat, or Triticum aestivum, which is the opposite of that, which is a high yielding but low quality type of wheat. So here we've got an example of durum wheat on the left, common wheat on the right. Is it easy to tell them apart visually? Yes, with these cultivars it is, especially at this level of magnification. We've got the longer, slenderer grains on the left of the screen there from durum wheat, and the shorter, fatter grains on the right with common wheat. But what happens when we have a mixture? Well, by using multispectral imaging and building up a model, we can effectively look at this on a pixel by pixel basis, and the multispectral imaging device will attribute different colors to the different pixels depending upon the likelihood of the grains arising from Triticum durum or Triticum aestivum. And here, the blue grains, or the blue colors there, are indicative of a greater likelihood of the grain arising from Triticum durum than Triticum aestivum. And again, we've been able to optimize this approach further. Here we've got an example of 10% adulteration in a Petri dish there. We can effectively remove the background using multispectral imaging, and within 20 seconds, once we've built up a model, effectively screen this to tell which grains and which pixels are more likely to arise from particular grains as well. So it's a very effective and simple screening approach there. A second example I've got here is in the herbs and spices area, and we're looking at oregano. So we can build up a spectral profile based on oregano in a Petri dish there. We can also look at other materials as well, such as myrtle, which will produce a different spectral profile from oregano, olive leaves as well, which again will produce a different spectral profile from oregano. And when we've got a mixture in there, how do we know it's oregano, or is it composed of other things? It looks a little bit like oregano, smells like oregano as well, but with multispectral imaging, we can quickly determine if there are aspects or ingredients in that sample which do not meet the spectral profile typical of oregano. Now, why is this important? Again, I'll defer to one of our other centers of expertise who are part of the panelists today as well, who were involved in some of this as well. But in 2016, the FSA's Food Crime Unit, as part of their annual strategic assessment, commented on an oregano sampling exercise in 2015-2016 that revealed that around 25% of samples looked at contained gross adulteration from olive or myrtle leaves. Now, again, I've given an example of authenticity here, but actually multispectral imaging can also be used not just for authenticity, but for potential quality purposes. So here, in our first example, now we've got a petri dish of oregano. The spectral profile there, you can see there are red bits in there as well. Now, if, for example, we did a DNA test on this oregano, it will come out as 100% oregano. But we built the model up on the actual leaves of oregano. And what we're seeing there in terms of the red are the sticks and stems associated with oregano as well, which you may not necessarily want in your oregano herbs and spices shelf. So again, it's a nice simple screen to check, is there something in there that shouldn't be in there? So moving on to my third example there, ground almond shell in cumin. So we've got some ground cumin there, builds up a nice spectral profile, some almond as well, which obviously is powder format and produces a very good and unique spectral profile. What about when we've got a mixture? It looks a little bit like cumin, but visually there may be something in there. Again, using multispectral imaging, we can quickly identify if there's something in there that shouldn't necessarily be in there based on the original profile. Again, why is this important? Well, as Sterling was saying earlier, in 2013, we had the horse meat crisis. In 2015, we had various RASAVs, Rapid Alert Systems for Food and Feed, in place, which alerted us to the fact that there may be potential almond or mahaleb in spices labeled as cumin and paprika, which resulted in a number of global recalls of that. And again, almond being one of the relevant allergens that requires labeling according to the relevant directive. Another example here, meat species identification. We've got some blended beef in a petri dish, some blended pork in a petri dish as well. And when we've got a mixture, may not necessarily be visible by eye, but using multispectral imaging, we can determine which parts of the sample are more likely to be beef and those which are more likely to be pork in origin. My final example for today is ground peanut in ground almond. Again, we've got some ground almond in the petri dish there, built up a nice spectral profile, one for peanut as well. And then when we've got a potential mixture there, is there something in there that shouldn't be in there? This is 75% almond, but with 25% peanut present, which visually you may not be able to see, but multispectral imaging can pick that up for you. And again, why is this important? This is because I think in 2016, a restaurant owner in North Yorkshire was prosecuted for manslaughter following them providing a takeaway meal to a customer, and the customer said they were allergic to peanut, but the takeaway meal contained almond, but the almond had been substituted with the lower grade peanut in there as well. So just to finish off then, why is multispectral imaging useful? Well, as Sterling said earlier, looking at the analytical process, we can look at various modules going from sampling through to analysis, all of which take time and cost money. If we're able to go from sampling straight through to the instrument, this could potentially save us on some of the time and efforts involved. So multispectral imaging, once you've built up a model, is very rapid, 20 seconds or less. It's non-destructive, you'll get the sample back. It's non-targeted and a multi-analyte analysis as well. You only need the one instrument to look at a variety of food samples. And there's minimal consumable cost there, just the cost of a petri dish as well. So I'm conscious of the time, so I'll just flick over to the last couple of slides. So why is multispectral imaging useful? Well, I've given some examples of where it's been used, but it's also can be used in rice analysis, grain analysis, detection of nuts, coffee beans, Arabica versus Robusta, etc. And it's really then not to replace pre-existing methods, but to augment the analytical toolbox for food authenticity testing, and can be really used as part of a triage system, screening for the presence of potential issues prior to the analysis of confirmatory genomic or proteomic approaches. So if you'd like to find out more, we participated in a number of DEFRA and FSA projects. You can type in the keywords there into Google or go along to the DEFRA and FSA websites or even the Food Authenticity Network website. And also we use multispectral imaging in the government chemist program and some of the analysis for innovators projects as well. So just to thank all those involved, including the Food Standards Agency, DEFRA, LGC, government chemist, and also Gavin Nixon, the science leader within my team. And thank you very much for your attention this afternoon.
Thank you very much, Malcolm, for that fascinating look at actually seeing some results. I know as analytical chemists, we're used to seeing peaks, but sometimes it's nice to have some technology that actually looks at some samples and gives you some images. Thank you very much. I'm sure you'll have got questions for Malcolm. Again, please drop them into the Q&A chat, and we'll catch up again with Malcolm in the Q&A session. Thanks very much, Malcolm. So it's my pleasure now to move on and introduce our third speaker today, who is Dr. Christophe Noel, who is the lead for SGS UK's Food Authenticity Centre of Expertise. Christophe is a Health and Innovation Director at SGS UK and has over 15 years experience in the food testing industry. So with that, I'll hand over to you, Christophe, to talk about next generation sequencing, next generation solutions and challenges. Thank you, Christophe.
Thank you very much. So yes, today I'm going to speak about NGS and the challenges it brings to all of us, really. So the idea of the introduction of molecular in the food industry is really not new, but actually not giving a full kind of course of history. All the application we've got in our field now are originating from two major breakthroughs from Sanger in '77 and Carey Mullis in 1983, who invented basically sequencing for one, Sanger, and the PCR for Mullis, and both got Nobel Prizes for this. And the introduction of PCR in our world came quite quickly in the 90s, and we're all very familiar with PCR, real-time PCR for testing GMO, for example, meat, fish speciation, but also detection of pathogens, for example, in the food industry. And we're a bit less familiar with sequencing because it was deemed quite expensive, and it kind of got revolutionized in the early 2000s when capillary system and fluorochrome were brought from academia to our world. But just to illustrate how expensive and lengthy it could be, the human project was completed over 13 years and cost something like £2.5 billion, well, dollars, which puts it at about $1 per base, because an haploid human genome is about 3 billion bases. So you can imagine if you want to do any of those technology in our field and you want to characterize microbial organism, bacteria mainly, you think, well, it's 3 million, it's 5 million bases, it's going to cost me 5 million and take me 10 years to do. So not really, but it was very cumbersome. And in 2005, next generation sequencing became a reality. And why is it so important? Why has it been so revolutionary? Well, because when we had Sanger, we were having to sequence either one single organism, or we had to be relatively targeted. And if we wanted to do a whole genome, we had to be really clever and put little bits of DNA in plasmid and do a lot of constructions, library, etc. That was really lengthy, very complicated, needed a lot of expertise, and it was very long and costly. And then the introduction of NGS arrived, and that was a real true revolutionary, because we were capable of dealing with complex samples, and we were able to actually look at matrices that were much, much more complex. We were not thinking about one organism, or we were not thinking about looking at specifically to just one mixture coming from one organism. We could do everything at once in a way. And that was amazing, because we were kind of suddenly opening up a brand new field of application in the food industry. And when we talk about NGS, we're talking about a tool, okay? We're talking about technology. And next generation sequencing as illustrated in this slide, there's a lot of different technology to do it. We all heard about Illumina, PacBio, ONT, 454, which is now obsolete and it's completely defunct. But basically, all these technology are capable of delivering some application. And those applications have got really cryptic names sometimes, like WGS, whole genome sequencing, metabarcoding, metagenomics. But actually, out of all this, what can we get and what can we do to address questions in the food safety, the food quality, or the authentication? And I think that's where the difficulty is most of the time, is we've got a whole of a lingo and some very kind of complex words that are a bit daunting, and we don't always understand where we could apply them. And if we could start with just the food system, and that could be a production plan, it could be a new product, but it could be also us in a lab being confronted with certain questions about what you have on this plate, or am I dealing with my salmonella or real salmonella from my customer? All these questions now can be addressed using next generation sequencing. And it's interesting because we can use those very complicated words and give them a real meaning and applications. So when we talk about metabarcoding, for example, well, what can we do with that? It's usually using targeted amplification of genes. And what we are doing is looking at mixed cultures. So we are looking at potentially probiotic, a biofilm, all these kind of very difficult environment and community of bacteria that were really difficult to decipher and to deal with. So we can do this, and we can do it with an amplicon sequencing approach, which is metabarcoding. We can push it a notch and do what we call metagenomics. So here we're looking at the whole genome of all these organisms in a certain context. And what we're looking at then is potentially metabolic pathways, or how do all these organisms cooperate, and what do they have as a genetic pool, and what kind of capability do they have, and how can they do what they do in their environment. And this is really important. But the one I want to focus a little bit more is the whole genome sequencing here, because this got a lot of application in a service lab like us, but also for our customer who starts to hear about those words. Whole genome sequencing, or UKHAS is doing some, HSA, sorry, is wanting my isolate to be sequenced fully using NGS, and they want whole genome sequencing. What does that mean? And how is it done? And really, what is it going to tell me? And that is really daunting, because knowledge is power, and knowing what it really means is very, very important. And that's where we can help, putting some meaning behind all these words. So when we talk about whole genome sequencing, what is it used for? Is it really the holy grail of the molecular investigation? Well, whole genome sequencing means we're going to read the whole genetic material of an organism and try to understand what it is, what it can do, can't do, and get as much information possible about this organism. And what can it be used for? Well, we use it for factory mapping, tracking, and tracing. For example, I found an organism somewhere in my drain, for example, and I find it into my product. Is it really the same organism? Okay, so it's cross-contamination in investigation. You can do this in a lab as well, okay? Is the strain that I find into a product really theirs? And is it been there all the time? And what kind of property does it have? Or is it my last QC strain that has jumped in? And then there's also the linkage with the outbreak. And I think that's where the big names and the big brands are really concerned, because now we can sequence a salmonella and look historically, is it the same organism that has actually been involved in a previous outbreak? And that is really daunting. And understanding what whole genome sequencing and what are the implications of it can have is obviously very important in that instance. So when we do a whole genome, what kind of information can we actually retrieve? Well, we probably heard of multi-locus sequence typing, which can be done using Sanger, but you can do this obviously with whole genome, because when you do multi-locus sequence typing, you're looking at only usually 7 genes, about 1,000 of the genome of the beast. But it's usually completely sufficient to create some good linkage and have a good picture of what's going on. But sometimes you want to know more. Do you really want to know if isolate 1 is the same as isolate 2? Not that they're highly related. So what you then do is you need to read the whole of the genome. So you start by, when you say it's Salmonella enterica Nottingham, you just read the title of the book. When you do multi-locus sequence typing, you go a bit further. You're reading about 7 chapters, just about, at least the first page of each chapter. And then it gives you, okay, I'm looking at 2 books that are really, really similar. But are they the same edition? Are they exactly the same? Well, you need to do a whole genome sequence typing, because then what you're doing is you're reading the whole book, and then you can say, yes, those 2 copies are identical. Or there's only very few typo, and they're highly, highly likely to be the same. Okay. And then that's what you can do using that too. So you can do MLST, whole genome, MLST. You can do SNP, single nucleotide polymorphism. When you look at the typo in the book, and then you just say, well, there's that many typo, or there's so little, they are the same copy. But you can go a bit further. You can look at serology, for example. It's very difficult to do serology. Not a lot of labs do have serum nowadays. So you can do this by just looking at the genes that are involved in that that markers, on those markers, and tell you. You can look at antimicrobial resistance, virulence factor, etc. But the real question is more data always beneficial? Okay, and that's where we really need to start the questions. Is, okay, how do we do whole genome? Of course, it's sequencing, but there's a lot of steps in that sequencing. We start with a culture. So we need to make sure that isolate is pure. And then once we know it's pure, we need to make sure that we have the right quality for the DNA to be processed, because this is what we're going to read. We need to make sure it's exactly the right state and not contaminated, but also in the right amount, etc., etc. And then obviously for the sequencing, we're going to have to do a lot of different processes, notably create what we call libraries. So put bits of DNA in a certain context, you can sequence them. And that is very important, because you could lose bits, you could add bits, over amplify certain bits, etc., etc. So when you're going to do your sequencing, you're going to also look at the yield, what we call the yield, the amount of data you're going to retrieve. So that's the first bit. That's kind of the wet labs place. So you kind of look at already all these steps and how many QCs you need to introduce. Then you've got, once you get all these data, and we're talking about a huge amount of data, gigabase, and gigabase in terms of space on a computer, but also gigabase in terms of DNA. You just generate gigabase of data. So you need to assess how many reads that means. What we mean by that is how many little bits of DNA you've read to actually generate all this. Do you have enough? Do you have too many? Are they in the right place, etc., etc.? The coverage, how far deep do we go and have the right amount of data? And also what is really important, the software choices. How do we actually make this data meaningful? So the real challenges are we all talking the same language? Are we having the same references? You see, there's all these words I've used: genome, core genome, pangenome, core genome MLST. Are we using always the same words? How is the data obtained? Okay, well, is there some sort of consensus, some standards? Yes, there is. We started to have some clues on what and how we should do it. You know, big organizations like the CDC with PulseNet, IFST, UK, HAS starts to issue some guidances, but there's also some ISO standards that are helping us to actually format and makes us all talk the same language, have the same kind of references. So service lab are coming in. They need to do it the right way. Otherwise, we're going to compare apple and pears. Who is holding the data? Are we doing the curation? Are we going to share this all together? Or is it proprietary to the company? So it's about knowledge transfer and retention. And what is really important is what we call the metadata. It's all the information that is associated with the sequencing itself. So it's always going to be there. So when we talk about challenges, yes, whole genome is rapidly becoming the new gold standard for investigation, but we need to have good rules for standardization. They are being developed, but they will have to be constantly reviewed because this is a very fast evolving technology. We started in 2005 and we're already on generation 3, 4. So ultimately, the interpretation was always rest on the metadata. What, where, when, and how. And context will always be everything in a meaningful interpretation. And I hope I haven't been too far out my timing.
Thank you very much, Christophe. I was going to come in, but you thought I could see you. We're on a flow, and it was very exciting. Actually, NGS and whole genome sequencing is very exciting technologies. But thank you for relaying some of the huge challenges we have in this area. It's not unique just to these areas. It's unique. It cuts all the way across food authenticity, as we know, but some real issues that we will need to grapple with in the scientific community, and hopefully translate that in a way that's palatable for the food industry, who are our customers. Thanks very much, Christophe. And anybody, I can see already one question for you, but we'll come back to that in the Q&A. Thanks, Christophe.
Thank you.
So moving on to our fourth speaker today is Dr. Terry McGrath from the Food Authenticity Centre of Expertise at the Queen's University Belfast. Terry is a founder and the Chief Technical Officer for Analytical, and before joining the company, full-time, Terry was a research fellow at the Institute of Global Food Security at Queen's University Belfast, where his latest research was on developing spectroscopic methods, both lab and field deployable methods in conjunction with chemometric models to detect food fraud. So with that, Terry, I'm going to hand over to you to explain what all that means. But the title of your talk is Spectroscopy and Chemometrics for Authenticity Analysis. What does that mean? Thanks, Terry.
Thank you very much. Okay, so I will do my absolute best to simplify what I think spectroscopy and chemometrics is about and how it is used in food authenticity analysis. So my name is Terry. I have two hats I wear. One is a visiting scholar to Queen's, and the other one is a CTO in the Analytical. So I've been in Queen's for probably 12 years now, working as a part of Chris Elliott's research group in the Institute for Global Food Security. So the Institute for Global Food Security is really well respected around the world, not only being established as a Centre of Excellence in 2022, but in terms of the university grading scales, we are ranked #1 for lots of different things, including the REF 2021 return. We have a nice new building, which you see in the bottom left-hand corner that we work from. So in 2020, we formed via Analytical, and the vision of via Analytical was to become the world leader in developing the future food quality, authenticity, and safety analysis. I'm not going to go through all of this slide, but basically 2013 was a catalyst for the work that we were doing on authenticity with the horse meat scandal. It ultimately led to the ideas of forming the Analytical, which was then spun out as COVID hit in 2020, and then we had a launch event in 2021, and we're now offering 18 different tests for different herbs and spices for authenticity testing, of which 2 are accredited, and we're currently in the process of speaking with UCAS to get the other tests accredited. So now that stuff's over and done with, we go on to what I really want to talk about. What is spectroscopy and chemometric modeling? So spectroscopy is the study of the absorption and emission of light and other radiation by matter. What does that really mean? to me, what it means is spectroscopy is a study of how things interact with light. So what this actually involves is splitting light down into its individual wavelengths. So that picture on the top left-hand corner, you've got white light going in, going through a prism, and all the light is getting split out into its colors, so into its wavelengths. And where I work on this infrared region. So it's beyond the visible region. So the infrared region is where I'm working, using a device called a FTIR in this instance. So what you have here is a very quick example of a spectrum for mint. So along the bottom, you have each of those individual wavelengths or wave numbers for FTIR. And then up on the y-axis, you have how it is interacting with the light. So you get these nice little fingerprints that can be unique for particular foodstuffs. And this is what we work with to do our analysis. So chemometric modeling, next challenge, what is that? This is a very long sentence and it took me forever to try and figure out what it means. It talks about maths, statistics, formal logic, and all sorts of things. But to me, what chemometrics is, it's a tool that's used to extract the relevant and useful information from spectra that help tell you things about your samples. So spectroscopy and chemometrics actually has a lot of use. Can be used for food authentication, can be used for proximate analysis, so your protein concentrations, your oil concentrations, applications for shelf life, applications for quality control. Is this powder the same powder as I produced last time? Is this product coming in the same as what it was the last time? It can be used for safety applications, for process control. So if you're a brewer or a cheese maker, you can check to make sure things are going along and hitting their maximum yield. Likewise, there's also traceability applications. A few years ago, I had a look at the scientific literature in terms of authenticity testing using spectroscopy and chemometrics, and it covers a wide range of food stuff. That was way back, that was probably about 5 years ago. I looked at this information. Now, there is a big difference between what's in academic literature and what is actually commercially available and possible. Maybe something we can discuss during Q&A. So, a use case. This is spectra of oregano and adulterants ran on a near-infrared device. Now, maybe one or two can pick out certain samples and say, oh, they're different, they're definitely adulterated, or there's definitely something funny about that sample, but I can't. So that's why I use chemometrics. What you have on the right-hand side here is the exact same data. So those spectra have been processed through the chemometric algorithms to give us this score plot on the right-hand side. And what that's done is it's broken out oregano samples and what I've called not oregano samples. It does this by looking at those features in the spectra and trying to group together as best it can the things which are similar, and then trying to push it apart from the things that it's different from. So that's why we end up here with this is a two-class model with two clusters, one of oregano and one of not oregano. So what that means whenever I come to do my test is I'll analyze your sample, and if your sample falls in the blue area, like that little red dot there, I'm pretty certain that what you've sent me in is authentic oregano. It's no different from the other samples. Now, if I test your sample and it appears over in the green area, like this red dot, I'm pretty certain that what you've sent me in is absolutely not oregano. It is adulterated. It could even be purely adulterated sample. Things become a little bit more challenging then. So I analyze your sample and it comes between the two groups. So what that to me indicates is that your sample is a mixture of these two components. And in some instances, this is a linear relationship. So the more of the adulterant you get present, the further it moves towards this green group. One final scenario to talk about is if you get a sample which I analyze and it comes down here. So what that's telling me is that that sample is adulterated with something, but it's not anything that I've used in my models. It's something totally different and it needs to be further investigated. So Sterling talked earlier on about these handheld devices that are going quite low price and stuff, and how easy they are to use, which is true, but the important thing is that you need to have really good science behind the building of your models. So your models are only ever going to be as good as the samples you use to build them. It's really important that you have authentic, traceable samples, I would say supplied by industry, or at least have undergone the full industrial process, because if what you find is if you mill a sample in a mill, in a laboratory, it doesn't represent the same process as what it goes through in industry whenever it's working on ton levels or whatever they're working on. Models need to be robust. So they need to take into account things like country of origin, different producers, the acceptable species that's allowed to be present, seasonal differences, differences in production processes around the world, and many, many other things. One of the things about these types of models is it is possible that they may only be suitable for a particular stage in the production. But the most important thing to understand whenever you're working with spectroscopy and chemometric modeling is that validation is key. You need to make sure your method is well validated using spiked samples, using incurred samples, all forms of samples that you can use to prove how well your method is working. There are pros and cons of using spectroscopy and chemometric modeling. One of them I've already mentioned is it's untargeted or non-targeted. So we're not actually looking to detect a specific substance. That's why we can see that sample that was down in the bottom right-hand corner of that score plot I was showing you, because it's not actually targeting any particular compound. What in reality we're doing is we're modeling what authentic looks like. So if it sits outside that blue area or a perimeter around that blue area we set, then we know that the sample is not compliant. Really fast turnaround times, maybe not as fast as 20 seconds for FTIR because we have to do some grinding. But if push comes to shove, I could turn you a sample around in less than 30 minutes if you really wanted a result that quick. I've already said it models what normal looks like. Once the model has been developed, it is really simple to use. So I firmly would agree with Sterling in terms of the NIR. If a good model has been developed by the scientists and you put the testing instrument in the hands of non-scientists, you can get just as good a result. There is limited or no sample prep needed, and already mentioned the potential for portable applications. In terms of cons, I can't always tell you what the problem is. I can tell you there's a problem. I can tell you your sample doesn't look normal, but I can't always tell you what that problem is. Your sample collection and validation are really critical. So if it also takes a long time to develop a method. So yes, if you send me a sample in, I can analyze it in 30 minutes. But if I haven't got that method developed, it could take me a year to develop that method, depending on sampling. People view these models as black boxes, so people don't trust them. And the final thing I want to say is that it's very much based on percentage scale. So percentage adulteration. Is this 10% adulterated, 20% adulterated, not down to the ppm, ppb level. And actually that is, I'll leave this up for 2 seconds. So if anyone wants to take down my details, I'm very happy to speak to anyone about what we do or talk more. But what I'll do, I'll hand you back now to Silverani.
Thank you very much, Terry. And all the speaker details, I think, will be provided at the end of the webinar as well, if people want to contact the individual speakers. Thanks very much, Terry. It's really fascinating. And yeah, notice that validation is king. I think that goes for a lot of the techniques that we've spoken about today. Now, before we start the Q&A session, I was just going to ask Robin to put up the results of the poll that we asked earlier, where we asked you how many of your organisations have a written food analysis strategy, and the answer was 40% of the organisations had a written food analysis strategy, whereas 60% didn't. It's quite very interesting. As somebody who works in a laboratory, I would have expected the written strategy to be higher. So maybe we can come back on the Q&A and touch on that. So let's just kick off the Q&A session then, and I'd like to invite our first speaker, Sterling Crew, to join me to co-chair the session. Welcome, Sterling, again. I see that we have a number of questions in the Q&A. Did you have any comments on the poll initially? Now, can I invite the rest of the speakers to turn on their cameras as well, please?
I'm surprised the other way that so many do.
Okay.
As a retailer, I've been into thousands of factories globally, and as a manufacturer responsible for a supply chain, I've been into lots of manufacturers globally. And though all organizations do analysis, they have to by law. It's a legal requirement. It's often a customer requirement, and it's good practice. But in my experience, actually, and I put myself in that camp in some of my early work, I didn't have a written strategy that was part of my HACCP risk assessment process embedded. I had, and what I've seen a lot is QC strategies. And so it's interesting to get that. So it shows you that we are maturing as a profession and we're getting better. I think that's dramatically different than maybe I've seen 5 years ago. So it's great news that people are aware. Maybe this is a self-selecting audience, that because the people who might attend this might be a bit more aware of the issues because they have a passion about it. And that might be why so many have it. But it's really great to see.
Thanks very much, Sterling. We have a number of questions and about 10 minutes left for the Q&A. So maybe we'll take the most popular one. And the first one is, what is metacoding? And I think that's directed at Christoph.
Yeah. So metabarcoding, here we go. We are back to a weird word. So it's a good way to investigate populations of bacteria, fungus, or any kind of population in one go. So basically you select a ubiquitous marker, a gene that is basically shared across a population. Typically, we talk about the small subunit, the 16S, the 28S, for example, for fungal and bacterial. And then you just basically go in, amplify the whole lot in a community, gather that information, and next generation sequencing is capable of taking that information, which is a huge mixture, and creates little entities and little bags of data, and tells you this bag belongs to E. coli, this one belongs to Acinetobacter, etc., etc. So it's a good way of investigating a population, like a biofilm or a probiotic. Do you want to know what is in your probiotic? Well, you need to go in, find a gene that is capable of grabbing them all, and you can read the whole data and just say, here you go, I've got A, B, C, D. So it's using a little bit, an amplicon, rather than reading the whole book again. I hope it helps.
If I may add to that as well, because we also at LGC do some DNA metabarcoding for plants and animals as well as sort of microbes as well. So from our angle, exactly as Christoph said as well, but it's really sort of next generation sequencing combined with DNA barcoding as well. So you've got the DNA from our end, DNA sequencing of the universal genetic markers together with a reference barcode database to allow taxonomic identification. But yeah, I agree with what Christoph has said. Thank you.
Thank you both for that. I'm glad you two answered that question rather than I tried to field that, so that's good. Let's have a look at some of the other questions. We've got a question here. With all the significant developments in detection capability, what similar advances have been made with sampling? A very good question. Before I open it up to the panel, I was just going to say that we have a cross-government joint knowledge transfer framework for food standards and food safety. And one of the priorities I identified for this year was an e-seminar on sampling. I'm not saying that that's going to solve everything in the world, but what it would do is it will lay out some approaches that the Food Standards Agency use for different types of sampling exercises. The sampling is for surveys as opposed to homogeneity testing, etc. So that should be available towards the end of this financial year. That's what I'm going to say. But there's a related question on sampling again in terms of what are the best sampling techniques to ensure that you've got a representative sample as well. Sampling is a huge topic. I don't know if anybody wants to have a go at answering that. Don't all rush forward first. I'm going to pick on one of you if you don't.
I would just add there's a British standard for it. So I can't remember the number now. I'm not very good. I'm getting older, but there is a standard that makes sure that you have that your sample is statistically significant and involves rolling dice and things like that to make it very random. And there are lots of codes of practice on how to take a sample. To me, it's more important than the actual analysis, because if you get that wrong, you really have a problem.
I think you sort of hit the nail on the head. Sorry, go ahead, Malcolm.
Sorry, yeah, just to complement what Sterling was saying there, I think sampling is key as well. We've seen some slides today with the analytical process. The sampling is the upstream sampling process is critical. It doesn't matter how accurate or precise your downstream analytical technologies are. If you're not providing a representative sample, then you're not potentially going to get the right result out. As analysts, we are confined with and we are restricted to receiving the sample in the laboratory environment, and we can only work with that sample. So following the relevant sampling regulations as well, I think is important. So for example, with GMOs, there are specific regulations that we follow for sampling of GMOs as well. But I think it is a universal issue, and the key thing is the representativeness of that sample that you receive for the analysis. Thank you.
Thank you, Malcolm. I think, you know, the panelists have hit the nail on the head. It's not one size fits all. I think depending on what you're trying to do the sampling for, you need a different sampling strategy, and that's probably not what people want to hear. But unfortunately, that's the reality of the situation. And there are, as Malcolm says, some legislative sampling that you have to do for GMOs, things like mycotoxins. But there's a huge raft of other analytes that there's no sampling guidance for, other than sort of generic British standards, which tell you sort of good practice. I think sometimes people ring you up and say, well, how many grams of sample should I take? Where in my bulk should I take? And unfortunately, that sort of practical sampling doesn't exist. But the seminar that we're hoping to use does have some sort of statistical basis around how you should choose different sampling strategies. So these will be a first step, I think, to try and put something out there, and then hopefully we can build on that. There's lots of other questions coming up. There's one on I think for Terry. Could any of these spectroscopic techniques be practical for on-site use by factory production staff in a deboxing stroke decanting room?
Yes, 100%. There is lots of device manufacturers out there who have devices for use inline, online, at line, near line. So all of these applications are available. All these devices are available. It's about getting the applications built for those devices. So yes, it's technically possible, but it will take money because a lot of these things are going to be based around your specific commodities. So it takes a bit of bespoke model development. But yeah, I've seen this type of thing happening in the animal feed industry or cereals and grains. Just starting. Yeah,
Thanks, Terry. And I think, yeah, some of these technologies are really exciting, but I think, as we said before, based on the reference material, it sort of says, is a sample different? So if the food industry operator had what good looks like, it would tell you that it's different from that sample, but it may not necessarily tell them what it is.
No, but if you're interested in things like oil content or moisture content or
Yeah.
protein content, then it'll you can match it into your wet chemistry.
Yeah, yeah. I think it depends on the application. Sterling,
Yeah.
did you have your hand up?
Yeah. Mind if I ask Terry like a follow-up question? I'm really interested in the citizen scientist. We've seen how the Internet of Things has enabled people to understand what's more in their food. Terry, if I had an allergen sensitivity, and you know how important it is in legislation and for businesses, do you ever see a day where I could turn up, have my meal, use NIR or something, and just look at the food and say, hey, yeah, there are nuts in there or not? So I don't have to relying on labeling in a restaurant or cross contact. Is that a possibility for the future?
I think that's the would like, the tricorder idea would like. As I said in my presentation, at the minute, we can get down to percentage levels. We can't get down to ppb, ppt. So if you have an allergen, if you're very sensitive to a particular allergen and it just takes one particle, then it's not going to be detected at the minute with NIR. Some allergens like milk or liquids will be much easier to detect because they will be the sample will be more homogeneous, whereas things like nuts, it may be in pockets in the food. So to cut that very long story short, in the future, if they can get the sensitivity of the devices up, then I can see it having applications in certain food types.
Malcolm, go ahead.
Yeah, just to add to what Terry's saying and Sterling's very important question there, I think, again, with these, imaging devices, we've got examples of inline ones, portable ones as well. To agree with what Terry's saying, we're really looking at in terms of sort of percentages rather than parts per million, I think, for detection. And again, we've spearheaded some projects with the FSA and DEFRA using imaging devices and validation of those as well. And I think all we've been talking about today as well, it's not just unique to, for example, imaging devices or NGS. The analytical approaches we're looking at are very much dependent upon sampling, but also these reference materials or databases. And that's a key requirement all of the time as well, and equally well to show the fitness for purpose in terms of use on a production line, etc. As we've discussed, looking at method validation, the generation of objective evidence to show that a method is fit for its originally intended purpose through assessment of these important key performance characteristics, which is one of the slides that Sterling showed earlier on today as well. And again, to enforce that as well, looking at certification to ISO 9001, and for method-specific applications, where appropriate, accreditation to ISO 17025. Thank you.
Thank you, Malcolm. That was a really comprehensive wrap up for that question. I'm conscious that it's 3:17 and we started the Q&A a little bit late. Are the panelists okay to hang on for a couple more minutes? We can take a few more questions and then I think we'll wrap it up. Just to finish on the allergens point, I think given what Terry said, if you're hyper allergic to some of these food proteins, one molecule could be enough. So I think with that in mind, I think this idea of, having an app on your phone, you can scan your food and it will give you a big tick, you can eat it, I think would be very dangerous to do at the moment for food allergens. It's not, I know there are some products already being marketed to consumer facing applications, but I think until the validation, representativity, all of those issues are sorted, and then even if you've got all that sorted, you've got these hyperallergic people. So I think it'd be very difficult to have that application for allergens and be, hand on heart, 100% satisfied. Right, I'm going to take the next highest voted question. That was, does the light intensity affect the food composition in terms of soluble nutrients of food like vitamins? Terry, have you seen that?
Sorry, ask the question again. Does the light intensity...
Does the light intensity affect the food composition in terms of soluble nutrients of food, like vitamins? So I assume is it damaged or?
Well, for multi-spectral imaging and things like that, it doesn't damage the sample, so it's fine. It's not going to damage it in any way, shape, or form. But for FTIR, it's a destructive technique in that we have to grind the sample. So it's it's not going to be used as a food product again.
After-
That particular sample, I think it's probably maybe just bad practice for something that goes into the laboratory to go back out as a food, something that's food related. But for NIR can be used, and it does not. There's no sample preparation for some applications there, and the light, it's standard light. You're just breaking out the wavelengths, and you're using those. So they're using standard bulbs. Well, maybe that's standard laboratory type bulbs. So it's not going to, as far as I know, impact any nutrients unless you have a very, very light sensitive nutrient, because you're only exposing the food sample to these things for milliseconds at a time. So on the scale of things, I would say it shouldn't have an impact, or it won't have an impact.
I suppose the question was around the online use or point of use. So, you know, not the laboratory-based testing, but if you had it in a factory or on a line, could it affect those? Malcolm, did you have anything to add to Terry's answer?
No, not really. Just to reinforce such things as multispectral imaging, hyperspectral imaging, and for example, near infrared, you're just doing a quick snapshot, just exposing it to literally milliseconds of that, and it's not a prolonged exposure at all. So yeah, it's very unlikely to have any material impact upon the actual sample itself. Thank you.
Okay, thanks, Malcolm. And I think we'll just make this the last question and then wrap up. Very interesting to learn more about the tests which can identify food fraud. I work for an SME food manufacturer whose responsibility in the wider food industry is it to test ingredients for authenticity. I don't think this would be feasible for us to do regularly. Interesting question. I think I'll start off the answer, and please do all chip in. I think in law, it is the responsibility of the food business operator to produce safe and authentic food and put that on sale for the general public to consume. So yes, I know the SMEs have huge challenges in order to comply with the raft of legislative requirements, but ultimately the responsibility does lie with the food business operator. Anybody got anything to add to that?
From my side, I would also look at it as, don't view this as a chore, view this as helping to protect your brand identity. If you get caught up in a food fraud event, even through no fault of your own, it could cost you contracts that you've worked hard on. So it might need just a change in thinking around what actually is the purpose of the test as well. It is there to protect the consumer, but it's also there to protect you and your brand, something that you can stand over with the people who you supply to and say, no, we have tested our product and our product is good.
And it doesn't have to be
out, Silverani, that
go ahead.
The legal position, which is a due diligence defense, that you took all reasonable practical steps, and the requirements on an SME, and I totally understand where the question is coming from, a fantastic question, are different than if you're a multi-global business. And I think it would be reasonable if you're a small business that you could say that you had confidence in your supply chain. There are simple, basic, practical, common sense things you could do. But if you're a small business, the defense you put forward would be completely different than if you're a large business. And this is a very common question for SME.
And it's a valid point Sterling made there, because testing is only one part of the arsenal that you can use to protect yourselves.
Yeah, so supply chain assurance. It's the whole thing from farm to fork, really. But it doesn't have to be onerous, and I think this is the reason that we asked the poll question, that having a strategy, you know, having something in place, it could be just annual testing, it could be supply chain assurance, etc. So it doesn't have to be one thing. Just wanted to quickly mention that you're not on your own as well. There's lots of resources that Sterling mentioned in his chat. There's resources in the resources section of this webinar. And if you look on the food authenticity and food fraud prevention section, there are guides specifically for SMEs that will help you put in place a food fraud prevention strategy that's palatable for your budget and for the appetite within your business. And there are also initiatives like the Food Industry Intelligence Network, where you can collaborate with other parts of the food industry, and the analytical data is pooled on an annual basis. And I think currently they're looking at 5,000 data sets a year. So you can actually learn from the testing experiences of much bigger players and then adjust your testing strategy accordingly. So there is help on hand, but if you have any more follow-up questions, please do contact any of us and our details will be available shortly. So with that, I think we better wrap up. We could be here all day. You're talking to analytical chemists about analysis, and we could go on forever. But I'd like to thank you all for attending and thank our speakers for such interesting and engaging presentations. And in terms of, there's been some questions about the sharing of slides. So the webinar has been recorded and including the Q&A, and that will be shared with everyone who attended today via email. Will also be placed on the online webinar hub of the IFST and on the events page of the Food Authenticity Network. Before you log off, please can you fill out the short survey, which will launch shortly after the webinar ends. I'd just like to thank everyone again, our speakers, and everyone for attending today. And a special thank you to Robin for all the behind the scenes work he's done to make today run and be successful. Thank you very much and enjoy the rest of your day.